Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
I really want to thank my co-organizers. I did nothing except tell them that they were crazy that they could get a workshop of this caliber done so quickly. They thought of it about 8 weeks ago, and they mailed invitations to an incredibly distinguished list of people, and I said, there's just no way that these people are going to say yes on, 6 to 8 weeks notice. And all but one of the people did, so we're just incredibly excited. And this is also the first workshop of a new project that we are running, which is part of a program that Tony will tell you about in a couple of minutes. I've got just a few administrative details. You should pick up, if you haven't already done so, you should pick up your name tag back there and the schedule. If you want to go inside the building, one of us from Microsoft has to escort you in and get you a registered name tag so that you're allowed to wander around in the building. So if you want to do that, please get in touch with one of us. The restrooms are that way and then turn to the right. And also, if you have a car parked here, please go to the front desk and register your car at the first break, because we don't want any of your cars ticketed or towed. Okay, so we are very lucky to have Tony Hay open this conference for us. Tony is the Corporate Vice President for External Research at Microsoft. He is the person who is reaching out not only to the CS community, but to the biology community, the physics community, the chemistry community, and we're just very, very lucky at Microsoft to have Tony. So he will open this meeting for us.
Jennifer, thanks for your kind words. Yes. It's great to be here, and it's very good that you've come at such short notice, and I'm deeply impressed that Christian and Jennifer have managed to get you here for what I hope is a very exciting meeting. So I should just say a few words of background. I used to be a physicist many years ago. I came to the States in 1970 as a postdoc in particle physics at Caltech, and I had a couple of Nobel Prize winners in the group, Richard Feynman and Murray Gell-Mann, which made it, shall we say, an interesting time. But then I became addicted to computing and to parallel computing in particular, so I was ran a research group in parallel computing and head of computer science. And eventually I got lumbered by being Dean of Engineering, which I managed to escape by running the equivalent in the US would be the NSF Cyber Infrastructure Initiative. So I ran the UK's eScience Initiative from 2001 to 2005. And one of the reasons I joined Microsoft was that I became convinced by the amounts of data that scientists were having to deal with, from sensor networks, high-throughput devices, satellite surveys, all sorts of deluge of data from many different devices, that really we were moving into a new era of science. So you have theory and theory and experiment, but also now computation, simulation, of course. But I think that now, if you like, arguably, there's research that you do on data that maybe you didn't collect, and you put together data sets and you analyze them and you combine them. And so this requires the traditional skills of a computational scientist, but also, I think, a new set of skills for analyzing and mining data, and new techniques for doing that. And I was talking to the Director of Science at DOE, and he said, oh, well, Jennifer Chase and her group have a wonderful technique for doing these things. So, of course, then I found out that he appointed Jennifer to her first job in the UCLA. So I became less impressed after that. But no, so I think there are really interesting things, and I don't know if you know, but this document is from Stephen Emmett's group in Cambridge, 2020 Science, which illustrates in the same way that a new set of technologies and techniques are going to be relevant to the way we do science in the future. And so I think bringing together computer scientists, the IT industry, and scientists to try and solve problems that people care about. And that's what I came to Microsoft to do. And for me, it's a wonderful job. I work with wonderful scientists, and I can pretend to be a physicist or an engineer or a computer scientist, depending on who I'm talking to. I also meet very wonderful computer scientists in Microsoft research all around the world. My inspiration was Jim Gray's work, who sadly is no longer with us, but Jim Gray worked with the astronomers and introduced them to the idea that there was something beyond file systems. And actually, if you had relational databases, you could do different things. And he built one of the first web services for actually analyzing and combining huge amounts of astronomical data. One of our first activities in my program I set up in Microsoft was with David Heckerman, who used data mining tools originally devised for identifying spam in email, but actually identifying complex genetic correlations in HIV/AIDS. So he's working with HIV/AIDS researchers. So roughly speaking, the program that we're trying to put together in Microsoft globally is to attack problems that people care about, all right? So that can make a difference. And I think that Microsoft technologies, both the computer science research technologies, but also some of our product technologies, I hope can make a difference. And they have to coexist in a world, I know, of open source, of Linux, of Macs. We understand that. And people will only use our technologies if they're any good. So I'm assuming that we can demonstrate that they are actually of some use. So I think it's reasonably altruistic. I think it's really exciting. And I think the possibility, our vision for the future is that you'll have a platform which you can build up with, commercial stuff from Microsoft, from our products, research tools from people like Jennifer and Christian, from our research environment, open source tools that the researchers love and know, Linux clusters, Windows clusters, Oracle database, SQL Server databases, a whole heterogeneous mix, but that our software does something useful that scientists want to do. And so our vision of the future is that, if you like, an e-research platform for doing this distributed data-centric research. We'll have software on the server and the desktop, but also you'll have scientific services delivered from the cloud. So software plus services is the Microsoft's vision for how the IT industry will go, and I think that vision is actually going to be appropriate for the academic community. Already you're seeing services like from Amazon with their S3 simple storage service and their EC2 elastic computing in the cloud. I think we'll see services like this in the future from lots of people. So I think it's a really exciting time. I think that Microsoft could do huge amounts, can genuinely help the scientific community, and what we need is feedback from you as to what we should be doing, all right? So as an example of the publishing part of the phase of, first of all you have to get a problem, then you're going to analyze the data, and you have to do all those things, but then you want to write it up. And we have not done anything to assist people to write up. So you could, for example, imagine that you're submitting your great work to Nature, and they send you a template. Well, you could actually have a plug-in to Word which actually says, oh, it's to Nature, and it automatically puts it in the format for Nature. It automatically puts the tags that Nature want and that you want, and it automatically extracts the metadata so you can deposit it in a repository as well as send it off to Nature. All sorts of things could be done, require your ingenuity. So I hope you'll have a great meeting, and I hope you have lots of debate. Give Jennifer and Christian a hard time. That's what you're supposed to do. And I'll try and drop in from time to time. But really enthusiastic, and I think that really it would be nice if Microsoft could really play a constructive role in helping you solve some of the major problems around the world. So it's not just the biology, which we're doing here, but it's environmental science, it's in fact carbon sequestration. In China, the problem they're looking at, south of China is flooded, the north of China is desert, so they want for 3 months of the year to divert all the rivers to the north, potential for great disaster, so it would be nice to know what the effects are. So we're looking at problems which are regional, but actually people care about. So if you have ideas, then by all means talk to Jennifer, Christian, and myself Don't promise we can do anything, but we promise to listen. Okay? So I hope you have a great couple of days. I'll try and drop in, and you can always send me email, tonyhay@microsoft.com. And you might like to look at our website, Science at Microsoft, www.microsoft.com/science. And again, we would like to make that an interesting site and a community site to go along with it. So we're just beginning, so please bear with us, and if you have suggestions, please let us know. Any other questions? People's happy? I hand over to you. Yes. You only have a certain number of...
Microsoft can only afford 2 microphones.
Very good.
We're giving the rest of the money for basic science, okay? So our first speaker this morning is Dan Gusfield from UC Davis, and I love his title. It's Recombinatorics. That's the first time I've ever heard that word, but you know, I think it's a great word. Recombinatorics, combinatorial algorithms for studying and exploiting the history of recombination in populations.
Being a first speaker, though, I wasn't sure of the interests of the audience. So I'm going to actually talk about 2 different topics. One is more mathematical and algorithmic on that side, and then another one on the more biological side. And both of these topics are in what's called population genomics. And basically, that's the era we're in right now. The human genome sequence, it's a draft sequence, but it's basically done. And what we would like to do, or people would like to do, is to sequence many more individuals and correlate the similarities and differences in their sequence with differences in their genetic traits, diseases or disease susceptibility or a whole variety of traits. This is a week old, so it says presently we can't sequence large numbers of individuals, but we can sample. So I'm going to be talking about sampling. I say this is a week old because I think maybe 2 weeks ago, there was talk of maybe resequencing 200 individuals. Then I heard there's talk of resequencing 1,000 individuals. Just recently, I heard Craig Venter saying he's going to resequence 10,000 individuals. So this may not be true for much longer. But at the moment, we can't resequence many individuals. And the resequencing of Venter's diploid genome was a big accomplishment. That was just a couple of months ago. So this data that we deal with, I say we can sample what's called SNP data. Now, SNP stands for single nucleotide polymorphism, and that's just a place in the genome where you have 2 different nucleotides in some frequency in the population. And because of that, we can just think of this as binary data. So if I took a look at all the individuals here and looked at some particular place in the genome, we probably would be identical with that one single nucleotide. But roughly every 1 per 1,000 or maybe 1 per 500, and it differs in different regions, we'll see some differences. And the differences tend to be just 2 different variants. So even though there's 4 possibilities in any residue, any nucleotide, typically in a population, if you see any variation at all, you see 2 variants. And that's very, very useful for people who like to think about binary things. So our interest is in reconstructing the histories of the derivation of the binary sequences that we see today. The simplest model, which is the nicest mathematically and the one that we build off of, is the following perfect phylogeny model. So you assume there's some ancestral sequence. Here I'm representing it as all zeros. This is just an example of 5 sites. And then throughout time, each one of those sites mutates exactly once in the history. So here's where site 1 mutates, and the sequences below that have a 1 in position 1. Here's where site 2 mutates, and the sequences below that have a 1 at position 2. So in SNP sequences, the general assumption is that if there's been any mutation at all, it's happened once in history. And we're talking now about a single population, say humans or other organisms. So we're really talking about very recent history. So this assumption of one mutation per site is valid, and it's the assumption that the population geneticists take and call the infinite sites assumption. So these sequences that are here can be derived on a tree, perfect phylogeny, with one mutation per site. And so this becomes a kind of an explanation for how these sequences that we see today evolved historically. Now, as I said, I didn't know for sure what the interest of the audience is. At the very end of this talk, if I get to that part, I have some motivating explanations for why we want to reconstruct these histories. But right now, I'll just say this is a large undertaking by both biologists and computer scientists and mathematicians. All right, but getting back to the clean mathematics of this, when can a set of binary sequences be derived on such a perfect phylogeny? This is a necessary and sufficient condition that was found many times independently. If you think of the sequences arranged in a matrix, which is the way I had them, and let's just assume no duplicate columns, the sequences can be generated on a unique perfect phylogeny if and only if no two columns, or call them sites, contain all the four binary pairs, 0, 0, 0, 1, 1, 0, and 1, 1. And this is, in population genetics world, called the four gamete test. So each one of these binary pairs is called a gamete. And sites that fail the four gamete test are called incompatible. A site that's in no such pair is called compatible. All right, so for example, the black sequences here are the ones that I had a minute ago. And if you look at any pair of columns here, excluding the red, you'll see that this has a 0, 0, a 1, 1, 1, 0, 1, 0, but it doesn't have a 0, 1. So that pair passes the four gamete test. And if we look at every possible pair here, we'll see that every pair passes the four gamete test. And that's what allowed this perfect phylogeny for the black sequences. And the theorem says that this tree is unique. But when we add this red sequence, now we've added in the 0, 1 in this 4, 5 position. And the theorem says there is no such tree that can derive this full set of sequences. So this is both a problem and an opportunity, because mutations don't really capture the full set of biological operations that create the extant sequences. Probably the most important other operation in terms of a given population is meiotic recombination. And so that operates in the following way. You have two binary sequences, and what's generated is a third one called a recombinant sequence, which consists of the two sequences, the parental sequences are the same length. It consists of a prefix of one sequence followed by a suffix of the other sequence to create an equal length third sequence, the recombinant sequence. And so this P says that this sequence is contributing a prefix, yes, this is contributing a suffix, and this is the crossover point where the contribution moves from the prefix to the suffix. Okay, so this is a recombinant sequence. And now, if we allow recombination like that, we can generate all of these sequences. This is the tree I showed you earlier. And then this is the recombination I just showed you, and it generates that new sequence, the new red sequence. Okay, so now if you think about it, I should say it in a minute before this slide, but if you think about it, with one mutation per site and enough recombinations, you can generate any set of sequences. So there's no longer a question of feasibility. There has to be some objective function which says which particular networks are most biologically meaningful. So we have a whole variety of results. This is the menu, starting from clean mathematical and algorithmic results, and I'm going to tell you one of those from part one. And then we have practical computational methods that can efficiently compute lower and upper bounds on the minimum number of recombinations needed, and that problem of computing the minimum has biological validity. Various applications to biological problems. Gene conversion is one specific one that I hope to tell you about, because that's the other part of this talk, which is a more applied, and then this is a recent new model of recombination. But I'm just going to tell you two results out of that menu. So we're interested in minimizing the number of recombinations in networks that derive binary sequences. Again, we only allow one mutation per site, and that's combinatorially what makes the problem interesting. But also it's biologically valid in this context. There are certainly contexts where that's not a valid assumption. here's the basic problem. You're given a set of binary sequences, M. You want to find a phylogenetic network, that's one of the objects I just showed you, that generates M, starts from some ancestral sequence, and you have the mutations and recombinations, which are generating sequences. And you want to minimize the number of recombinations that you use, or recombination nodes that are in that network. And again, you only allow one mutation per site. So there are special cases of this that have polynomial time solutions. Rich literature that is called galled trees. We did that from 2003 till even till today. There's some new results. But I'm going to not tell you about those. I'm going to tell you about unconstrained, those general networks. And the minimization problem is NP-hard in general. So that's the backdrop. So what can we do? We can solve small data sets optimally with exponential time methods or on algorithms that actually work well in practice, a number of those kinds of results, or efficiently compute lower and upper bounds on the minimum number of needed recombinations. And in the data we've looked at in various contexts, these are close, so that's useful, and that has various biological consequences. And then apply those methods. And again, I'm going to show you an example of that. So the first part, the part one, is the mathematical clean result that I'm going to tell you about. Since minimization problem is NP-hard, the general motivation is to break up a problem into subproblems that can be solved separately and then combined. And the decomposition theorem really gets at the heart of what can be done along that direction. And the key tool turned out combinatorially to be useful over and over again is just a crude representation of the data. So here's the original data that has violates the 4 gametes. And we have one node. We build a graph here called the incompatibility graph, which has one node for each site. And then 2 nodes are connected by an edge if and only if the pair of sites are incompatible. So if you looked at 1, 4 here, you would find all 4 gametes in there. And so this is the graph that crudely, well, it reflects the incompatibilities, but you have obviously lost the raw data. Now this incompatibility graph turns out still to be very informative for things that are going on in the data. In particular, the connected components are very informative. Actually, it's typically the non-trivial connected components. So, for example, this theorem, the number of non-trivial connected components in the incompatibility graph is a lower bound on the number of recombinations that are needed in any network. And it turns out to be a fairly useful lower bound as well, and better than the first lower bound that was generated by the biologists in 1985. But I'm not going to talk about that. I'm getting to the decomposition theorem. So we'll need the following concept. In a phylogenetic network, if we look at a recombinant node, a recombination node, we can trace back 2 paths. There may be choices. But ultimately, because in a phylogenetic network there's only one root, those 2 paths will meet, and that defines what we'll call a recombination cycle. Okay, just a little bit of notation or terminology. And if I find in a network a recombination cycle, it might be sitting off by itself, or it may be sharing an edge with some other recombination cycle. And therefore, we can find a maximal set of these intersecting cycles, which I call a blob. So in more graph theoretic terms, if I remove all the directions on the edges, a blob is just a biconnected component of the network. Okay. So if we take each blob, each one of these biconnected components, well, everybody here knows this kind of thing. If I contract each blob to just a single node, then what I get is a directed rooted tree. If I didn't get that, that would violate the maximality of one of those blobs. This is a very simple thing, but it's a nice way of viewing networks. So I can think of any phylogenetic network as a directed tree of blobs, which we call a blob tree. And the tree is, of course, the blobs are the non-tree part. The tree backbone is like that. So this is my cartoon of the way I want to think of any possible network as a tree of blobs. All the ugly stuff is happening inside of a blob. Otherwise, we have this nice tree. And this earlier work on galled trees was just the special case where each blob was just a single cycle. And that's what we started out with in 2003, and after understanding that, moved on to these blob trees. So here's a very simple observation. In any network, N for M, all the sites from the same connected component in this incompatibility graph must appear together in a single blob. You can't split sites from a single connected component in the incompatibility graph into two different blobs. It just doesn't work. It violates ultimately some of the incompatibilities that were assumed to connect these in the same connected component. Well, this is a simple thing to prove, and we'll need that for the decomposition theorem. So the decomposition theorem is really about the other side of this, which is that for any set of sequences, binary sequences M, there is some phylogenetic network that derives those sequences. It has one mutation per site and has recombinations. But the blob structure is particularly nice. Each blob contains all and only the sites from one non-trivial connected component of the incompatibility graph. So there's this nice one-to-one correspondence between the sites that are in a non-trivial connected component of the incompatibility graph and the sites that are in the blob. And the compatible sites can always be put on edges outside of any blob, so they can be part of the tree structure. And we call this a fully decomposed network. And it's the finest decomposition possible in this term because I already told you that two sites from the same connected component cannot be separated onto two different blobs. So if we have one blob for each connected component and then the blobs are connected by a tree, that's as fine a decomposition as is possible. So here's an example. This has one blob because these two cycles are connected together, share an edge. But this is the same set of data generated on a fully decomposed network. And you can see the connected component 1, 3, 4. Those sites are here, 1, 3, 4, and 2, 5 are down there.
Is it clear what happens inside a blob? Aren't there different ways that you can reconstruct it within a blob?
Yes, inside the blob is where all the ugly stuff happens. And because minimizing the number of recombinations is NP hard, we can solve inside of a blob with an exponential time algorithm. And that's why we want to decompose. So, yeah. Now what happens inside of a gall tree is when there's just a single cycle, and this happens to be a gall tree. Then we can figure out how to lay out the sites in there particularly easily in a polynomial time algorithm. And in fact, it just wasn't part of the plan here, but one of the really nice results from gall trees is that even no matter how many sites you have on a gall, the number of permutations, number of ways you can lay them out on the gall is bounded by 3. And that only happens if you have 2 sites, and if you have more than 2 sites, it's bounded by 2, and with another assumption that usually happens, it's unique. So gall trees are particularly nice, but at any rate. Now the backbone of the tree, the tree portion of this network, is invariant over all the fully decomposed networks for the data. And that backbone can be determined in polynomial time. So how you stitch together the blobs, that's easily figured out. Yeah?
When you restrict yourself to fully decomposed networks, are you losing optimality in some cases?
Yes, and that's what we're going to get to. That's the next portion of this. Yeah. That had been an open question. So the decomposition theorem was we first published this, I think, in Recom a couple of years ago, but the question of minimality or optimality was still open at that time. It's been solved, and I'll tell you about it. But the backbone part is invariant. So that means that we can find a network for M by solving the recombination minimization problem for each of the connected components separately with whatever tools we have available to us. And then we can combine those in an invariant way. So the combination isn't difficult. Okay. Well, Dick's question, I must have planted that. Yeah. While there is always a fully decomposed network, it does not necessarily minimize the number of recombination nodes over all possible networks. Okay. So that is, it sometimes pays to be devious and to push sites from different connected components together into the same blob. And but actually, well, I don't know if we can absorb this in real time. Here's an example of one where Yoon Song came up with this. The sequences in the original input are in black. This red sequence is something that was not in the input. The incompatibility graph for the black sequences has 2 connected components, and each one of them we can show requires 2 recombinations, so that would be a total of 4. And yet when you push them together in this single blob. You have three recombinations, and this is just a single blob. This also mentions something about a theorem that I'm going to tell you about. So unfortunately, yes, it's not good enough to restrict attention to fully decomposed networks if we're trying to minimize the number of recombinations. But we have several, some of them are natural and some are not unnatural, sufficient conditions for when a fully decomposed network, when there is a fully decomposed network that minimizes the number of recombinations. Okay. And the deepest one, this sounds a little technical, but actually gives a lot of insight into what's going on, is the following thing. So let's let N be some phylogenetic network for the input. And L will be the set of sequences that label the nodes of N. So at the leaves, we have the same input set, M. But internally, we may have some sequences, just as in the network I just showed you, we may have some sequences that are not in M. And if GL, we let GL be the incompatibility graph for that full set of sequences. Okay? Then the sufficient condition is that if GL and GM, M was the input set of sequences, if they have the same number of connected components, then there is a fully decomposed network for M with the same number of recombinations as in N. So again, we're seeing the role of this of the connected components. So yeah.
Can you find this fully decomposed network fast, or is that hard?
Well, you can find the tree structure, and you can find what the sites are that go into each blob. And yes, if all you wanted was a fully decomposed network, and you weren't trying to minimize. No, that's no easier. That's still NP hard. So the problem of finding the fully decomposed network that minimizes the number of recombinations, you still have that problem inside each blob. And so it's still NP complete. But if you want to find just some fully decomposed network, then that can be done in polynomial time. But here we have this theorem, which is not necessarily that applicable, but it gives insight into the structure of what's going wrong. Well, anyway, this is a sufficient condition for when a fully decomposed network has the same number of recombinations as the network we started with. And therefore, it tells you what you have to have in a counterexample. In an example, one that, okay, so just a way of emphasizing this, a fully decomposed network exists that does minimize unless every optimal network uses some recombination nodes or node that's labeled by some new sequence not in M, and the addition of those sequences creates an incompatibility between sites and different components of GM. Okay, so that, again, that tells you what you have to look for if you're looking for an example. And this, back to this picture, this has that property that the original data has two connected components. I haven't shown you what they are, but when you add this new sequence L and you look at its incompatibility graph, it only has one connected component. So the number of connected components has indeed changed according to the theorem. Now, there is a conjecture out there that what you save each time you push two components together is at most one. So there's some, which is what's happening in this particular example.
Bridging incompatibility between.
Yeah, right. That's another way of saying that the number is changing because it can, well, anyway, you're creating bridging incompatibilities across components. There are some more practical things we can extract from that sufficient condition. The following: if the data can be derived on some network N, which has the property that every edge contains at most one site, and every node is labeled with a sequence in M, then we can deduce that there is a fully decomposed network which minimizes the number of recombinations. So this is a much more practical one because you can imagine finding such a network that has that property, whereas the previous one was not. Well, here's another one, but anyway. All right, so that was the clean mathematical end of the menu of possible results. And as I said, that one is, it'll be published in Journal of Computational Biology fairly soon. It hasn't been published. Now, I want to go to the other extreme, not necessarily heuristic in the computer science standpoint, but heuristic in terms of the biological standpoint. What do we do with these tools that we have? So we have mathematical tools, we have algorithmic tools. How do we actually use those or encourage other people to use those to address biological questions? So this comes out of a paper, well, it's also about to appear, but it was earlier version was in Recom 2006. And this is algorithms that we can use to try to distinguish single crossover recombination, which is the kind of recombination I told you about, from another kind of recombination called gene conversion, which I'll define in a minute. So gene conversion really is just a recombination with two crossovers, at least in our terms. Biologically, it has all kinds of other meaning, and in fact, the name gene conversion comes from genetics before they saw the molecular basis of this. But anyway, in our terms, it's just that the two sequences create a third sequence, not by just prefix-suffix, but by the prefix of one sequence and then an interior piece of the other sequence, and then finally a suffix of the first one. So you can think of it as two crossovers. This also happens in meiosis, and it happens actually more frequently than single crossover. But the one thing I should say is this is called the track in here, and the track lengths tend to be small. So 300 to 1,000 nucleotides, although it's actually not totally determined what the track lengths are, and that's part of why this is not a well-studied problem. And when I give this talk to biologists, the gene conversion part of these talks is what they find most interesting, because it is a very understudied problem. All right, so gene conversion involves two short crossovers, and the track length, as I said, is thought to be in that range. And gene conversion, it might be the Achilles heel of fine-scale, it's called association or linkage disequilibrium mapping methods. These are methods that are very widely used now, and now meaning this last summer in particular. Yeah?
You said previously that the amount of SNPs, you have about one SNP every 500 base pair per 1,000, so that will make these regions have one SNP?
Yeah, that's another reason why this is, we haven't seen a lot of these things, because it's the density of the SNPs isn't enough to give you a lot of information about it. That's right. And that's, that's just true. All right, so association mapping or linkage disequilibrium mapping is a method, and if I have time, I'll tell you a little bit about it at the end, which is one of the motivations for understanding recombination and building these networks. But the logic of it all relies on single crossover recombination. And if there's gene conversion, it might really cause trouble, at least in the fine scale mapping where you're looking in small regions. Okay, so here's some quotes from people who know a lot more biology than I do. This one is saying, geneticists have typically ignored gene conversion, but it really can cause trouble in linkage disequilibrium, LD, which is the key to how you do association mapping or linkage disequilibrium mapping. And there's a nice book on coalescent and gene histories. Anyway, it came out a couple of years ago by Hein and Wolf, where they make this point too on page 211. They show how gene conversion can really cause trouble. Anyway, so that's just the background. Now let's just assume that we have methods, which we do and I haven't told you about, that can compute reasonably close upper and lower bounds on the minimum number of recombination events. And now I'm talking about either single crossovers or gene conversions that are needed to derive a set of sequences. We don't have practical methods that give the minimum number. In practice, it depends on the data. But in the data we've looked at, we can compute reasonably close lower and upper bounds. So how do we use those methods to say something interesting about gene conversion? And one of the interesting things that biologists have looked at is given data, it's SNP data, they want to know, was this derived with a lot of gene conversion or with very little gene conversion? Are there hot spots for gene conversion that are perhaps the same hot spots as a single crossover recombination, or maybe they're different? Or anyway, they just would like to look at the sequence data, the SNP data, and say, I think there was gene conversion and here's where it was. So how do we do that? So here's the naive, the top level idea. So for a given set of sequences, let BT be the bound, and we can either be a lower bound or an upper bound. In practice, we use both. That we can compute on the minimum total number of recombinations. So that's single crossover plus gene conversion when the track length is bounded to be T. I told you the track length is the length of the thing in the middle. Yeah. It's the middle piece.
Oh, the region that.
The one that's in the middle, yeah. So let me go back to that picture. Yeah, so this is called a conversion track, and its length, as I said, is short. But so let's just say we bound it in our computation. Okay. So BT is something that comes out of our algorithms. I mean, it's a number that's computed, but what it really means, it's hard to say what it means. It just comes out of our particular algorithms. So B0 is just the single crossover because we allow zero length, track length. And if our algorithms are already good, and they'll have this property, and they do, that that total number that we compute should decrease as T increases. As you allow longer and longer tracks, you should take some advantage of it and get a smaller number of events. And DT is just going to be the decrease in compared to not allowing gene conversion at all versus allowing gene conversion as long as the track length is bounded by T. Yeah?
If gene conversion in practice has about 1-2 SNPs per track, doesn't the approach of doing some pre-processing where you use look ahead, so you can go from left to right and check if the site is not compatible?
Yeah.
Then go ahead for 1 or 2 more SNPs, and you
Yeah.
have to go back to compatibility.
That is true, and I should have mentioned that there are there are methods that come principally out of the statistical world that uses those kinds of ideas. And so you will see positions that are separated by some other SNPs that look to be more concordant than the ones in the middle. And so you'll see those kind of deviations. Yeah. So all of this work is motivated by having a much more explicit picture of the histories, by trying to build explicit histories or having a concrete number like the minimum number of these events, rather than relying on those kind of statistical summaries. But you're absolutely right. You can do things like that, and those have been done. All right. So DT is the decrease or savings by allowing gene conversion as long as the track length is bounded by T versus not allowing it at all. And just for emphasis, DT is, again, it's not a property of the sequences. It's the sequences plus our algorithms. Yeah.
So when you vary your T, you have to process your data somehow, right? Your algorithm can compute the lower bound for a combination event given a set of sequences.
Yeah.
So when you change T, how do you change your input to your algorithm? That's something I don't quite understand.
The input to the algorithm is just a set of sequences, so we don't do anything different depending on T except run the algorithm with that as a... You're going to get a different number, yeah. So naively, I mean, whether we optimize this or not, naively you can just think of, as we change T, we just restart the computation and compute B of T, and then we know what D of T is. So there may be some slicker way of trying to save some of the previous computation, but conceptually, we have the input, which is just a set of these SNP sequences, and then for any fixed T, we can run our upper or lower bound or both, and find out what the minimum number, what our bounds are on the minimum number of operations.
But you don't know where the track is.
We don't know where the tracks are. That's right. We don't know where the tracks are. So I see if we had some notion of where they are, then we might be pre-processing, but we don't know. That's what the algorithm finds, is where it wants to do the gene conversion. Yeah. Oh, yeah. Okay, so, right, we have to translate from the real distance in nucleotides to the number of SNPs, and I have to punt on that one. I think we translate into the number of SNPs because that's ultimately what's being computed, but that's more in the modeling question of how you want to make these things realistic, or the computations useful. But I'll have to punt on that at the moment. Okay, so what naively do we expect from these computations? So as a function of T, so let me just give you the game plan. We generate data, two types of data. One that was generated with only single crossovers, recombination, so there's no gene conversion in how that was generated. And we have data that's generated with gene conversion. And we have data that was generated with different track lengths. It's given, the actual track length is given by a particular distribution. But imagine we have the two sets of different data with gene conversion and data without gene conversion. Then we run the programs, the upper and lower bounds, and see how they behave as a function of t. And what you would expect, or what we naively expected, was that the decrease, this is again the savings when you allow gene conversion, it will be a larger savings, and the savings will grow faster as a function of t when this computation is actually done for sequences that were generated with gene conversion and crossovers also, compared to when dt is computed for sequences generated with only single crossovers. So the naive expectation is that the algorithm that's computing these bounds on the minimum number of events, gene conversions plus crossovers, will be able to use crossovers somehow if the data was really generated with crossovers. Conversions, thank you. Yes, conversions plus crossovers, yeah. And we expect some convexity here because we're generating the track lengths under some distribution, and when you get past the mean track length under that distribution, you expect that allowing larger and larger track lengths in the computation, it stops helping that much. I mean, it's similar to the... so here's, again, a kind of cartoon of what we expect, is that if we had sequences that were generated with gene conversion as well as single crossover, and this is dt as a function of t, we'd see something like this. Letting longer and longer tracks happen in our computations helps to some point and then stops helping. And for sequences that were generated with only single crossovers, it doesn't really help that much to allow tracks gene conversion. And this is a slide that says what we actually do is more complicated than that, but I'm not going to get into the technicalities. The naive view is still, I think, informative for how you use these tools. And these are this is the summary of real data. And again, I'm not going to tell you the actual things we compute, but it's similar in concept and spirit to what I was telling you. These different plots here, graphs here, this is the curve for sequences that were generated without gene conversion at all. And this is the track length as it's getting bigger and bigger, and it fits our expectation. These other curves, we have one curve for each level of gene conversion that we simulated data. The solid curve at the top has the most gene conversion as compared to the other ones. They were generated with less gene conversion. But we're seeing the form that I was showing you, that allowing longer track length in our computations, upper and lower bounds, does help in decreasing the total number of events. And there's some kind of convexity. Now, these 2 points in there, that's sort of an artifact of the data. But anyway, the point here is that this basically conforms to what our naive expectation was. And therefore, it gives us a way of distinguishing. It gives us a signature for data that was generated without gene conversion versus data that was generated with gene conversion, and even some level of calibration for how much gene conversion there was. So the take-home message is that our upper and lower bound algorithms can't make up gene conversions. If the data wasn't generated with them, then our ability to use gene conversions doesn't seem to help much. Okay. And so everything I just told you was a lie. This is the slide that said it was all a lie. The slides, the graphs were based on averages. So at every data point, we did lots of data and showed you the average. But you need to apply this on a single data set if you're going to apply it to distinguish gene conversion. And unfortunately, what happened is the variance, these averages behaved very nicely. It provided those plots that I just showed you, but there's high variance. So if you want to apply this idea to a single data set, we have another idea called the gene conversion presence test, which all I'll say about it is that it works pretty well. This is the simulation data, and
So is this coming from the fact that there are hotspots for gene? Where is the variance in the real data coming from?
oh, the variance is in the computation.
Variance is in the computation.
Yeah. So Yeah, I mean, what I showed you here was the these are averages. I mean, if I take a particular t equal 1,000 here, then to generate this point, this is the average over many computations, many data sets that were generated with these parameters. And so the averages look nice, but when you want to apply this, you want to apply this on a single data set. And so you may not have this nice curve for that particular single data set. Yeah.
So you compute from your lower bounds, you compute those alphas for t and beta to t, I suppose, as test statistics for data.
Yeah.
They somehow summarize your data.
Yeah.
So you're saying the distribution of these test statistics under null hypothesis of no gene conversion overlaps too much with the alternative.
Yes.
With the distribution of the same test statistics under the alternative, which assumes both.
Yeah. If I was statistically literate, that's what I would have said. Yeah. But that's exactly right. Yeah. So, but there's a simpler test. We end up with a simple gene conversion presence test, which really under a two-stage optimization just ends up with whether this particular thing that we compute is greater than 0 or not. It is whether when we're trying to minimize, let me just paraphrase what this is. When we're trying to minimize the number of events, gene conversion plus gene conversion, in order to get that minimum, do we absolutely need to use some gene conversion? That's what this is saying. And if we get a yes answer for both the upper and lower bound, then that's our decision, that's our statistic then to say that, yeah, there was gene conversion in the particular region that we're doing our computation on. Okay, so just very quickly to say we have applied this to questions of gene conversion in Arabidopsis, and this was taken from data by a group at USC. Well, it says in press, but this has been published by now. And their problem was exactly what I've described. They were looking for signatures of gene conversion in Arabidopsis, and they had a fairly crude method. I can say that because it was ultimately came from me, for trying to detect gene conversion. And what I've been telling you about is a much more refined idea. And they found 4 fragments that they thought contained clear signal for gene conversion in that particular data. So we ran this method and we found 22 fragments under this particular gene conversion test. 3 of those, we found 3 of the 4 that they found, and the others, I'm not sure what the story is with the 4th. And on the others that we found that they didn't, there's no way really now, I mean, I don't know of any nice way of resolving who was right. But I would just say that what I've showed you is just a much more refined algorithmic idea. Yeah?
In 500 and 600 base pairs, you wouldn't expect many SNPs. Is that in units of SNPs or units of nucleotides?
That's probably SNPs. Yeah, I'm going to have to defer again to the particulars. But unless, well, I don't know. I'm going to say we'll talk about it later after I consult. All right, so you can find software and papers here. Now, I promised to say something about motivation for all of this. I think I still have about 5 minutes if I'm right.
Yeah, absolutely.
This is something I often talk about at the very beginning because this also represents some work of a graduate student of mine who finished. But it's a nice illustration of why we care about recombination. There are many reasons, but association mapping in diseases using ARGs. ARGs are what population geneticists, that's the term population geneticists use for what we've been calling phylogenetic networks. And it stands for ancestral recombination graph. Okay. Now it's actually, if you look at that, ancestral recombination graph is actually a stochastic process, but the graph that it produces fits what I've been calling this phylogenetic network, which is not necessarily what everybody else calls phylogenetic networks. It's unfortunate that there's too much of the same terminology. Okay, but here's association mapping, which, as I mentioned, last summer has been the summer for association mapping, where the results have been coming out hot and heavy. Previously, there were association mapping results that came out, but they were very unreliable. Now, it's felt that these are really finally working. So here's the basic idea from a networks perspective. This is not what the statisticians and biologists actually do. but it is what several are saying they would like to do. So here we have SNPs at these colored positions, and here's where some disease mutation occurs, and let's just say it's between the SNPs. We want to locate it. We want to know where's the mutation that occurs for that particular disease, and you name it and we can study it. So I'm going to give you an example of the simplest possible scenario, just to illustrate the idea. And in practice, things get horribly complicated because things aren't so simple. But let's imagine we have, it's not even a Mendelian, it's like super idealized Mendelian disease, where we assume that a sequence or the individual who has that sequence is diseased if and only if the sequence has that single disease mutation. So there's one mutation. If you have that mutation, you've got the disease. If you don't have the mutation, you don't have the disease. That's a highly idealized assumption. But now let's also assume that we know the true history of the sequences. Here's the phylogenetic network, which generated these sequences, Mr. A, person B, C, etc. E and F are diseased. Okay? So they have that, somewhere in the history here, the mutation happened that gave them that disease. So the two questions you can ask and you can answer is where historically where on the network did that mutation happen? So you can figure out it had to happen on this edge. This edge represents some time period. It had to happen here because we need a place where these two lie below it. So that if it was above here, then we would be getting, then C should have been diseased and so on. Now what we have below here is D, who's not diseased, and E and F, who are diseased. Okay? But the first thing we can figure out is that the mutation had to happen along this point in history. Then you can ask, well, why didn't D have the disease also? Well, here's a recombination. So now that gives you the hint as to where in the sequence it happened. Because wherever it happened, that was passed on to E and F, and here was a recombination, but F still got that mutation. So wherever it happened in the sequence, F still gets it, but D doesn't. And you can use that to figure out, to the extent that the data allows you, where in the sequence you get it. So if you look at what parts of this disease, of this sequence, did D, E, and F inherit? So you already figured out that the mutation is along in here. What part of this sequence do these three individuals inherit? You see, if my color coding makes any sense, that D gets the blue portion. It gets 3, 4, and 5. Yeah, position 3, 4, and 5 from here, but doesn't have the disease. On the other hand, E gets it directly. It gets the entire, without recombination. And F gets it, this beginning portion from this place. So you can deduce that the mutation has to be between SNP2 and SNP3. That's what's shared in common. No, then why isn't D sick? Well, SNP2, yeah, somewhere in here where, these are SNPs, but the actual mutation, the recombination between point 2 and 3 extends beyond there. The recombination is somewhere between 2 and 3. And so we know then that the SNP has to be between there, the mutation has to be between there, and it has to in fact be in the portion that D doesn't inherit. So you can get that conclusion. This is a very simplistic sort of thing, but there are a couple of papers in the biological literature which follow this game plan, and my student Yufeng Wu did this in a more computer science principled way with this style of association mapping. So this is called association mapping or linkage disequilibrium mapping. Dick, did you have a question?
How widely accepted is the assumption of at most one mutation per site? I've heard that it's not so widely.
Well, it depends on the context. So in the context of a single species, where you're talking about fairly small time periods, and where the probability of any mutation is small at an individual site, then the probability of 2 mutations at that same site, if there isn't some biological force that's making that happen, that's a reasonably good assumption.
That sounds logical, but I've heard it said that it isn't true necessarily.
Yeah, well, when I first got into this, it sounded like, oh, this is an assumption that gives us binary sequences, and I'm going to have to apologize for this forever because we obviously like binary sequences. But I heard talks like, for example, David Cox, who's a well-known geneticist originally from Stanford. Now he's the head of Perlogen, who's a company that does this kind of human variation data. His talk, he stated that as almost as an axiom. One mutation per site in the data that we're looking at. And I talked to him afterwards, and he just said, "No, God gave us that, and don't worry about it." And I mean that they have millions of dollars riding on that assumption. I can back off a little bit. If you see sites that don't seem to obey that, you can throw those out of your data. You're trying to see something that's happening in history, which is informed by these binary positions. And if you have some other sites that are doing something strange, it doesn't deflect from the overall picture. And I mean, necessarily, it depends on the question you're asking. But a lot of times, if they do see something that looks non-binary, then just throw it out of your data.
They might be strange because of recombination, or they might be strange because of a double mutation.
Because of recurrent mutation. There might actually be happening at those sites, but it's infrequent anyway, and it doesn't distract from being able to build up the picture you're interested in, which is informed by the binary data. So that's my present apology. Although I will tell you, I was here a week ago, not in Microsoft, but downtown Seattle. There was a conference on what's called copy number variation, which is now the next wave that's coming beyond SNPs. And those are non-binary. So I am altering my viewpoint to look at algorithms on non-binary data. And that's, algorithmically, that's, I think, a wonderful new way to go. Thank you.